calling I'm cool with whatever we do. Jioni safu no la with what we do. Hale yidemu new school. Nyepamne ne kudlukula. Nyepangi ragala gena. Nyepamne ne kudlukula. Ibuloke no moto ni beti sola. Ibuloke dato ni betoto.
Mingi Congo.
Lamin.
Na?
Lamin te jibe norin te nyamen Lamin.
Kabo na wulo kono.
Wulo kono nyinta mo Lamin.
Pabilenki.
Muna ngata afongole.
Haina.
Ngata. Ngoto le. Fabrama.
Mingi. Baba nyola.
Ngoto le Fabrama.
Namu jumara kan kuma ni ten.
Fabrama.
Jumara mu.
Ndele bare muso.
Nunda mana.
Kamo kata.
Jamani mbere.
Alindo ko.
Jamani.
Bari ngoto man kontande.
Bari jamani fana na mundu ma ko kontan.
Ngoto minaka ngalamin je fom filitala. Lamin jibe nori nyaala.
Lamin ila siran sama ne benki. Yala so tol sayni no ko ngol ko momo.
Ngoto.
Nam.
Itedinte yalo ne Lamin hakilo diata bake karamungoto. Anu santeng ministry ko di ngole si suwo ko amanke ko be holiday
si bitun
bagona ringa
ala ni ko kede namo balo
ala ni na ko na karta bulo be damna ala ni je
ngoto
nam
ngoto mon kontan feren
jamani mu ne keda
ite ka Lamin ki wulo ko no nyintay mon ko do fete malo Lamin nyande sila la be la moy kala karango
jamani karamol be soron ne ko fasita wulo ko nako
karamol ma soron ni ko karamol soron da amanke comme holiday lem ngoto dini ngo nyanta sila la ala karango la moy yamen kamen fue man kontan andamande
alla nyin kombo courant adanda la kombo len do la jankara mol le soron do kamen courant ma do la kombo e don dol fana na kara mol soron je kara mol soron Lamin ada lo nyino la aman tara fe na sokon ala mu nekela
Lamin ite fete malon ko lesinol be ke kar radio to
nga fine
e ni mo Lamin ya mu na fonye
kabim bina kan ntambita nteri maning Fatu tambide la sukala ala diningol bebe siri be la moyka
Faru danso am fana tambida jele ni subala la ngaje diningol be jet talanta nen kili warol e be seri jele
sa Lamin fanante ku kili wo ke no la ba Lamin bitaka wulo ko no nyinta mo Lamin
aman dara fenela
ite dine ko di man si yakari la suno la moy radio to e balon na
mu ne ka ke afonde ba nyara ni alon
telentel jikot
afonde ba mu de be ke
ite balon tel be jikritel ni man karambe ku kele yaje nata karana mbasa malaya e tarije
par ñarande kombo courant ho findira kombo ide na raje den sonka bay ko Lamin manya tala wulo ko karam mo be so da fa ñembe
ko to wole nati bayo ngalle ni Lamin kuntala kombo courant o ka fa me kombo courant asototal wala kinay ko e si suwo kono bari ni ko e si suwo kono be karangol di ke la radio lin telel to hani tele masoto to nyaay man tele soto da bungo bar radio be bulen koto batiri sanga ay si ay alamo
sa do kala
nyaadi ko ta o bulaji
ado kala nyaadi
kamun ke nyaadi
radio abe wala mo la be mona falaje
ibe la karandirol kala jele
ti talle be radio ko men ka karandirol
ila subjector baby jele baby jele
wala nan ka coco moy fa dan so ya ngom metambi ka nga dinni ngol je be ikadum bungo ko kada maro mo
asototal de
Lamin mala be karawala de
bari
laftalal ideal on ko inde area sakon wala fo
muna ni ma ko ndego folle sinila ba ndele sinilo ne ba
mba foke en koto wala fango yen wulindi yalo foke tabat ñu fana bulata ko ko me yen nati jambi Lamin nyanta karana ndel beji ko be nyineka ya bulay kara wulo kono ta kilar ya bambalo yo be bulaje koto dinni ngoy si ayala le sino la moy radio
wara ka munda kara mo man soro Lamin nyanta kara si so na regular
sila le ayala le sino
ndal ko korna ko smalo
kilar ya
mo be ngala ko momo sa mi do ni ko no kera
mengi bruzo
ndal ko la da ba rosa no radio ba no bari woro radio ni abe jele ba do ngamra do man dembe buljan dala nyara nyar wofram ko fendi mo la Lamin tayra ba rosa
folo ajibe nori nya Lamin tamete
teku
bari ko do famo ke
ja image nañu ñambol si zol le keje kisgi man si zol no le kafun ka ñowa bari ñambu kino ni mañana ka finde ni nambe ko do la ja
ngalle limbeta de be ñambo do dilanna
ñara nim ko ridin fennat ñara nata
ko bari famo ke da
ngata bungono tammi bungono ñara ngando ko fele ko ke baru nganda ni pelo la dija
iyo awa betar la
bungono bismillah bisu diara
pendulala ye fongo tanka sina dolu tanka koro ta koro ta
Good day, viewers and listeners. I am Samuel A. Mendy. I'm back again with another biology lesson. And the last lesson, we were talking about genetics and some genetic terms. And before I leave you the last class, I left an assignment for you. I said we are going to discuss it in the next lesson. So I'm here today, so we are back again to discuss that assignment. The first question was, what is monohybrid cross? What is monohybrid cross? We say a monohybrid cross, we said it's a cross between two individuals or two parents which differ in only one genetic character or involves in alleles of one specific character. That's what we said is a monohybrid cross. So the next question was, if the recessive gene responsible for skin color in albinism in humans is represented by A. So that means that the recessive gene is A, lowercase a for that matter. And it says, what are the possible genotypes and phenotypes of the offspring produced by a homozygous normal and a heterozygous albino? So here we are going to look at what the genotypes and we are going to look at what, when you said this a cross between the phenotypes, we are going to look at a cross between the phenotypes. The man is what? A homozygous normal. So when it's homozygous, and then heterozygous. Homozygous and heterozygous. Okay, so now this homozygous, the genotype is uppercase A for the homozygous, and the heterozygous is uppercase A and lowercase a. So what are the gametes? The gametes, you have A, and then A, uppercase A and lowercase a. So now we'll do what the cross. So when we do the cross, we'll find out that uppercase A crosses with uppercase A. The person is what? The offspring is normal. So this one crosses with this. This offspring is a carrier. So this parent crosses with this. You have what? A normal. And then this one crosses with this. We have what you call a carrier. Here, the question says, what is the possible phenotypes and the possible genotypes? So now the phenotypes, all of them, this is a homozygous normal, and this is a heterozygous, okay, it's a carrier. So now when you're talking about the phenotype, we have two normal, this and this one and this one, they are normal. That is two normal, a normal, and two, this one and this one are also carriers. So that is our question. So that means that two of the offsprings are normal. They do not have carrier of what? Skin color. They are normal. But two of the other ones are carriers of the albinism. That was the assignment, and this is the cross in the assignment. I hope we are able to get the correction. Those of you not get the answers, please look at the cross properly, and I hope the next time you will do better. Today we are going to look at back crossing, test crossing, independent assortment, and dihybrid crossing. We are going to look at back crossing, test crossing, independent assortment, and dihybrid crossing. So now, If you look at back crossing, we said a back crossing is a cross between an offspring in the F2 generation and the recessive parent. If you want to use a back crossing, I will take offsprings within the F2 generations. The offsprings in the F2 generation, we have not, this is just the F1 generation, this is the F1 generation. So we are going to come to the F2 generation. When you come to the F2 generation, I will explain how we do that cross. We said it can be used to find out the genotype of each offspring in the F2 generation. We are talking about back crossing. So you said it's a cross between an offspring in the F2 generation with a recessive gene. So like the offspring in the F2 generation, it may vary. The characters may vary. It might be homozygous, tall, heterozygous, tall, or it can even be recessive. These are the offsprings that you use, okay? These are the parent. We are using it to cross it with the recessive. So now when these gametes are given to us, how do we do the cross? So now we have this is the phenotypes, the gametes. We have T. These are the gametes. And then we are using the recessive genes to cross them. We used T and T, the lowercase t. This is homozygous tall, and this is recessive short. So now when you cross, what are you going to have? So T, uppercase T, lowercase t. And when you cross this, you'll also have uppercase T, lowercase t. You take this one, also cross. You have uppercase T, lowercase t, and also this one, you also have uppercase T, lowercase t. That means that for the offsprings, they are also going to be tall. If the character that we are looking at, it was the character for tallness and shortness. So that means that the phenotype is all tall, but genotypically is in heterozygous. So this is what you call background. You can also use this heterozygous to cross with the homozygous short. So that means these are gametes T and lowercase t. You cross with lowercase t, lowercase t. So what are we going to have as our offsprings? This will crosses with this T, lowercase t. This crosses with this, we have T, lowercase t. That's what we are doing is a back crossing using the Mendelian genetics. So you cross lowercase t, and then you cross lowercase t, lowercase t. So now what is going to be the ratio, the phenotypic ratio? The phenotype is going to be too tall, too short. So, and it's 50-50. If you want to look at the ratio is is 50-50. That means 1:1. So that means we have too tall, too short. And when you want to look at the phenotypic ratio is 50-50. So when you say 50-50, that means 1:1. So that means when you use a heterozygous and cross it with a recessive during a back crossing, you'll have the offsprings will have too tall and too short. And the ratio, the phenotypic ratio is 50-50. That's 1:1. So I will leave you to also do the other cross, the homozygous recessive. If it's a homozygous recessive, what are the offsprings that we are going to use? We said a test cross is used to identify whether a phenotype of an organism showing a dominant trait is due to a homozygous or a heterozygous genetic condition. We said it involved the crossing of the F1 with the F1 individual with the phenotype of a dominant parent with the recessive plant. And when you do the cross, what we just look at, that's what we are going to do. However, if the F2 individual is heterozygous, then the offsprings will show a ratio of 1:1. Like if it is a heterozygous, we show a ratio is 1:1. So that ratio 1:1 is also the same as 50-50. That means 50 are tall and then 50 are short. So that's what you call a backcross. If you look at here, this is what Mendel used, the flower of a plant. We have a purple, this flower is a purple. It is known phenotypically as purple, but genotype it is not known. And he used to cross with a recessive white flower. When this cross is done, what is the prediction of their offspring? So the flower, we don't know whether it is a homozygous purple or heterozygous purple. It's a purple plant. So he choose like what we just estimate, and then we came out with this. So now if it is a homozygous tall, this is where we have like this flower is homozygous purple. You find out that all the plants, all the offsprings were purple. That's the reason. All the offsprings were purple. And then if you look at a heterozygous, half of the offsprings are purple and half of the offsprings white. So in short, we can say backcross breeding an organism of unknown genotype with a recessive homozygous. You breed an organism with unknown genotype with a homozygous recessive. That's what you call backcrossing. So here we are looking at another law of Mendel. He talked about independent assortment. And then when Mendel was talking about independent assortment, he talked about when two or more pairs of contrasting characters are present. And then when you talk about these contrasting characters, you can say whether the plant was what, yellow and round. Whether it's a contrasting character, the plant, either it is yellow or round, or whether the plant is what, is red and having somewhat brown spot. There are two pairs of contrasting characters. You say each pair is inherited independently of the other. That means that if this character, whether it is round and yellow or yellow and round, when they are going to be independently assorted, one has to be, they have to be able to be assorted separately from the others. He explained that this law applies to independent pairs of genes, provided each pair of the gene is located on a different pair of chromosome. So now when you're talking about the chromosomes, we look at the different parts of the chromosome. And in the chromosomes, genes have a specific place. There are different types of genes, and those genes are located in a specific area in the chromosome. That's what we talked about, and we saw that. Likewise, also, it is independent assortment. Genes are located at a specific place in the chromosomes. So when they are located in that specific place, they will determine the type of organism that is available in that place. Whether the organism, whether it's talking about the height, or whether it's talking about the color, or whether the roughness, or whatever feature the plant, the gene is talking about in that area. We said, hence, the transmission of genes determine height and seed color are independent events. These ones, they are done independently. This means that the gene determination seed height are located on a pair of chromosomes, and the gene determined seed color are also located on another distinguished pair of chromosomes. So here, we are seeing the independent assortment. Like Gregor Mendel is talking about law of what? Independent assortment here. He said when gametes come together. So here, we are looking at a plant. We have SS, Y, Y. These are all, let me write it properly, capital Y, uppercase Y, SS. So that means that the plant is yellow, is having yellow seed and what? Round shape. That means the shape is round and yellow seed. Crosses with a green plant and having a wrinkle shape. So now we have what? Green is small case, SS, and YY. Okay? So now when these things cross, what will they have? So here, Mendel came up with, for the F1 generation, they'll have SY. This for the F1 generation. This is going to be their offspring. So at the end, we'll have about 9, when this phenotypic ratio comes, we'll have about 9 9:3:1. So how did you come about this 9:3:1? So you come about, you look at what gamete formation. How do we form these gametes? So now you have S, and then you reacting with Y. So now this and this will form, we'll have, these are the options SY, and then this one will also form with this, and then S, lowercase y, this and this we have S and uppercase Y, and then also have this S. Because of these gametes that we have here, we can use them and put them in a Punnett square. If we use this and put them in a Punnett square, we'll have the possible phenotypes of what the offsprings. So that's what Mendel did. He says, this is the female gamete, this is what the male gamete. Okay, so here I will put this how gametes are. So I'll put S, Y, capital Y, and then I put capital S, lowercase y, and then capital lowercase s, capital Y, and then small y, small s, small y. The same thing what I put as the male gametes, I also put as the female gametes. There's capital S, capital Y, and then capital S, small y, and then small y, capital Y, and then we have small s, small y, lowercase y. So now when you do the crossing, what are we going to have? This and this is going to be, the shape of this thing is going to be yellow and round shape. Then you have S, Y, Y. These are all uppercase. Then you have S, Y, lowercase y. And then you have uppercase S. This is lowercase s. Uppercase S, lowercase s. Uppercase Y, lowercase y. And then you have uppercase S, lowercase s. This is uppercase y, sorry. Uppercase S, small s, and uppercase y, and then small y. So this, we have to do all the crosses. We have to do all the crosses for this. And then we also do the same thing, uppercase S, uppercase y, and then have small y. And then also here we have what? Uppercase S, small y. Then also we have what? Uppercase S, lowercase s, uppercase y, small y. And then also have what? Uppercase S, small s, and uppercase y, small y. So all this cross, the round and yellow is 9. Round and what yellow is 9. That means S is equals to 9. Is equals to 9. And then round and green will have what? 3. Wrinkle and yellow is 3. So when you do all the crosses, you'll have what? 9, 3, 3, 1. That means 9 is to 3 is to 3 is to 1. If you do all the crosses, capital Y, then also have capital S, small s, capital Y, small y. Then we have small s, capital Y. We have also small s, capital Y, small y. Okay, so you have what? Capital S, small s, capital Y, small y. Then you have here capital S, small y, We have what? Small s, capital Y, small y. And then also have what? Small s, small y. So this is the cross that you will be able to find in the independent assortment of genes. So this was what Mendel was doing. So in that independent assortment, it goes with a dihybrid inheritance. You say in dihybrid inheritance or a cross, it's a cross between two different genes that differ in two observable traits. Mendel, in his experiment, studied the inheritance in a pea with two pairs of contrasting characters. He also explained the distribution in F1 and F2 generation of the two seed traits. That's color and height. Because like we are looking at here, this one is what wrinkle and green, or yellow and what round shape. These are the two things, yellow and round shape. This is yellow and round shape. We also look at what wrinkle and green. In an experiment, he crosses a pure breeding tall plant with yellow seeds with a short plant having green seeds and obtain 4 different kinds of plant in F2 generation. So that is the result. He said the result of the experiment shows that the appearance of all 4 possible types of plants regarding their traits are assorted independently in the phenotypic ratio of 9:3:3:1. That is the cross that you have just done, and that is a form of a dihybrid cross. So this was what I was explaining. So like if these things, if you are given this type of a dihybrid cross, so what you do, you just need to extract the gametes first. So like here, we are given another yellow and round flower. We are given yellow and round flower is capital Y, R and small r. Crosses with what also green and wrinkle flower. They crosses with what green and wrinkle flower. That's lowercase y, and lowercase r, lowercase r. So during the F1, their gametes, then we have uppercase Y, uppercase R, and lowercase r. This is their F1 generation. So now if you want to do this F2 generation, you need to extract what the gametes. So now when you are extracting the gametes, Y, uppercase Y, lowercase y will crosses with what? Uppercase R, lowercase r. So now when you extract this gamete, you can put them in a Punnett square. And then these are the gametes. You have uppercase Y, lowercase r, and then uppercase Y, lowercase r, and then lowercase y, uppercase r, and then you have lowercase y, lowercase r. So these are the gametes that you are going to put in what? In the male and the female, so that you can cross them. You will have what? The possible offspring. And the possible offsprings are going to be at 9:3:3, which is already plotted in the graph. So use this, like you put a Punnett square like this, and then try it, because this is the male gametes, and this is the female gametes. So you put this uppercase R, this, and then lowercase, uppercase R, lowercase, and then you have what? Lowercase r, y, uppercase r, and you have lowercase y, lowercase r, and then also come to the female side. You have what? Uppercase y, uppercase r. You have what? Uppercase y, lowercase r. Then you have what? Lowercase r, uppercase r, and then you come to what? Lowercase r, y, lowercase r. So use this and complete the Punnett square, because we have already done the first one. So please try and do this one for yourself. Gregor Mendel followed the character of the seed color and seed shape through the F2 generation. He crossed a true breeding plant with yellow round seeds with a true breeding plant with green wrinkled seeds, producing dihybrid F1 plants. Okay, these were the dihybrid, the F1 plants that we talked about before. And then he self-pollinated. He said he used this one and self-pollinated. and then he has a different, and then he has what? The different possible offspring. We said the two hypotheses, dependent and independent assortment, predict different types of what? Ratio. So this is another question that we are given. It says 100 tomato seeds, this is a sample problem. 1 tomato seeds were sown and the seedlings grown to maturity. The characteristics of the plants and the number of the plants of each type are shown below. Smooth pericarp with hairy stalk is 55. Rough pericarp with hairy stalk is 20. And then you have smooth pericarp, and this thing is 19. And then you have rough pericarp with smooth stalk is 16. It says suggest a genotype and a phenotype of the parent plant from which all these seeds were obtained. One of the things that you can do is you can possibly use the lowest value here and divide by each. Then you will have approximately the ratio. When you divide 6 by 6, you'll have 1. When you divide 9 divided by 6, you have 3, and then 3 point something. And then when you divide 20 by 6, you have 3 point something. And also when you divide 55 by 6, you have 3 point something. You just try that division. That is the approximately phenotypic ratio. But here, you can also come to that, to the calculation. So now, when you do this, we extract the gametes, like what we did here. We have smooth pericarp, S, and here stalk is H, capital H. Then you have rough pericarp, lowercase s, and smooth stalk, lowercase s. Parent genotype, S, that means uppercase S, double uppercase S, and double uppercase H. And then they react, the gametes fuses with what? The recessive. So these are the possible gametes, the F1 generation. And then when you do the cross, these are the possible gametes that have already been done here. So please cross-check it. And then when you do, this is going to be the smooth and hairy pericarp. And so this is where we are going to end. But before I make a leaf of you, let me give you this assignment that you discussed. You say a pea plant heterozygous for inflated pods, that is uppercase I and lowercase I, is crossed, that is heterozygous, is crossed with a plant homozygous for constructed pods. Draw a Punnett square for the cross. Assume pollen comes from the recessive plant. That means the homozygous recessive plant. B says pea plants heterozygous for flower position and stem length uppercase A, that means uppercase A, lowercase uppercase T, are allowed to self-pollinate. And 400 of the resulting seeds are planted. Draw a Punnett square for this cross. How many offsprings would be predicted to have terminal flowers and be dwarfs? Okay, so thank you. This is where our lesson is going to end for today. I am your teacher, Samuel A. Mendy. Bye.